So we're now thinking about enzymes. And an enzyme is a biological catalyst. And what a catalyst will do in chemistry is it will lower the activation energy for a reaction to take place. In other words, a catalyst will increase the rate of a chemical reaction. It will facilitate and increase the rate of a chemical reaction, making a chemical reaction more likely to occur. So enzymes are biological catalysts. They facilitate biochemical reactions. They control and regulate biochemical reactions. Activate, control the rate of, basically they govern all the biochemistry that's going on in the body. And you can always tell enzymes because they end in ase. So for example, a protease is going to digest proteins. An amylase is going to digest carbohydrates. A lipase is going to digest fats. But as we'll see, there's other types of enzymes as well. And enzymes are proteins. They're usually globular proteins in nature. And the key thing about an enzyme is it's not used up in the reaction. So this is true for a catalyst as well. So an enzyme or a catalyst, and we're thinking about enzymes, they are not used up in the chemical reaction. They are not part of the reaction. They just facilitate the reaction, regulate the reaction, increase the rate of the reaction. And what enzymes do is the enzyme will actually work on a substrate. So there'll be a substrate that's acted on by the enzyme, bringing about some biochemical process, and the end result will be a product. So the product is generated by the action of the enzyme, or the substrate is facilitated into the product by the action of the enzyme. So it's substrate into the enzymic system, product out of the enzymic system. Now enzymes are proteins. They're usually globular proteins. And proteins are long chains of amino acids, and they fold together into a secondary structure, and they fold even more times into a tertiary structure. Proteins have this complex protein folding pattern determined by the primary sequence of the amino acids. And what this means is that proteins are able to generate a molecular architecture. They have shape. They generate physical shapes with electrical properties, but they are nonetheless physical shapes. And enzymes have this physical shape that they generate as a result of this complex folding of the protein, and it's called the active site. So enzymes have an active site, and the active site is where the substrate binds into, undergoes its chemical processing to be released as a product. So this active site is actually where the activity goes on. It's the active site of the enzyme, this very specific particular shape. Now, the examples I've just given of enzymes are actually digestive enzymes. But there's two main ways we could look at enzymes in the body. There are the enzymes inside cells, the intracellular enzymes, and there's the extracellular enzymes. So throughout the body there are intracellular enzymes. So in each cell of your body there's a milieu of unbelievable complex biochemistry going on, with numerous biochemical reactions going on in different parts of the cell. And all of these are facilitated by enzymes. They are accelerated by enzymes. They are controlled by enzymes. Without enzymes, there is no life, because all this biochemistry that generates life is facilitated and controlled by enzymic systems. And actually, in clinical practice, this is quite useful, because when a particular cell dies, the enzymes that were inside that cell will be released because the cell membrane breaks open. So cell death, or even some cell injury, will release enzymes into the body fluids that were intracellular enzymes. And this is quite useful because we can do blood tests, look for these enzymes, and from that we can work out which part of the body is injured. So creatinine kinase might indicate a muscle or heart injury, or AST, aspartate aminotransferase, might indicate liver damage. Or there's quite a few enzymes that you'll come across in practice, but they are enzymes that indicate damage to particular tissues. Or sometimes we can even look for isoenzymes, because an isomer has got the same chemical formula but a slightly different structure, and there's different isoenzymes in different organs and tissues of the body to perform particular biochemical activity in that particular area of the body. But it's useful, so we can look for these enzymes as markers of particular tissue damage, because they've been released from the cells of that particular tissue when that tissue was injured, insulted, or indeed necrosed and killed. So it's quite useful in clinical practice. But extracellular enzymes, which we're getting around to thinking about, are used in the gastrointestinal tract. So these are usually secreted into the lumen of the gastrointestinal tract, where they are breaking down food components. And the aim here is to break down complex food molecules, which are usually insoluble. Complex food molecules to be broken down into soluble products, because we need soluble products. Basically it means smaller molecules which are water soluble, usually water soluble, some can be fat soluble. But breaking large molecules down into smaller molecules which can be absorbed into the bloodstream or into the lymphatic system. So most digestive enzymes are working within the lumen of the gut. Those on the enterocytes, the very final stage of digestion, are actually operating on the cell surface membrane of the enterocyte itself. So they're either in the lumen of the gastrointestinal tract or very close to the gastrointestinal tract. And that brings us on to another way we could look at enzymes. We could think about enzymes as being anabolic or catabolic. So anabolism, anabolic processes, are building small molecules up using energy into bigger molecules. That would be an anabolic process, as goes on, for example, in the synthesis of protein to make muscle, where we need to build up larger molecules. In this case, protein molecules from smaller molecules, which would be the amino acids. So that's an anabolic process. It's a building up, making bigger molecules. Some enzymes will facilitate these anabolic processes. Other enzymes will facilitate catabolic processes. Catabolic processes are to break big molecules down into smaller molecules. So the digestive enzymes we were considering are going to be catabolic, because they're breaking things down into smaller amounts, into smaller molecules, making them more soluble. Now, enzymes are very particular, very fussy, very specific, and they're very specific to the substrate that they can enzymatically convert into a product. So enzymes are substrate specific, and this is because the active sites that form a particular molecular architecture are a highly specific shape, and they have highly specific electrochemical properties on their surfaces as well. So they will only accept one particular type of substrate. So an amylase, for example, thinking about digestive enzymes, an amylase will only accept carbohydrates. It won't accept fats, it won't accept proteins. It will only break down carbohydrates into simpler sugar and disaccharide units. They're highly specific to substrate, but as well as that, they're highly specific to temperature. So at lower temperatures, the substrate, imagine that's the substrate there, and this is the active site of the enzyme. So the two have to come together. The substrate has to get into the active site of the enzyme. Now at lower temperatures, kinetic theory tells us there's less molecular vibration in a fluid. And as temperatures increase, the molecular vibration is going to become more intense. So that's a cold substance, and that's a warm substance, more vibrations. And the more vibrations there are, the more moving around, the more likely that is to happen, that the substrate will fit into the active site of the enzyme. So activity of the enzyme tends to increase with increasing temperatures, but then it reaches an optimum point because as temperatures rise, that's going to affect the chemistry of the active site. And as temperatures rise, the active site can be denatured. It can go out of shape, which means the substrate can no longer fit in because there's been denaturing of the active site as the temperatures become higher. So what we find is enzymic reactions get faster and faster as the temperature increases, then they reach an optimum, and then they can drop off quite quickly due to denaturing of the complex molecular architecture that is generated by the active site. And they're also very specific. Enzymes are very specific to pH, to the acidity, because the pH also has the potential to change the nature and the shape of the active site, and again alterations in pH can lead to denaturing of the active site. Now, it's probably well known that the pH of arterial blood is 7.35 to 7.45, so you'd expect a lot of body enzymes in the cells to work at that sort of those sort of pH levels. In the gastrointestinal tract it's slightly different. You get some enzymes working in the stomach where the pH is very low. You get some enzymes working in the small intestine where the pH is high. Low pH being acidic, high pH of course being alkaline. Or you get amylases working in the mouth where the pH is relatively round about neutral levels as well. So it's not that enzymes work at one particular pH, but specific enzymes work better at a specific pH, but that's dependent on the particular biochemistry of the enzyme itself. And indeed the rate of reaction can also be increased if there's a higher concentration of enzyme, if there's a higher concentration of substrate, and also it helps if the product is continuously moved as well. Otherwise the product tends to get in the way of the substrate fitting into the active site of the enzyme. So that's the little conceptual introduction to the nature of enzymes. Next we want to look at a little bit more about this interaction between the substrate and the active site, a little more about the specificity of enzymes, and then we'll need to go through the specific enzymes that have specific roles within the gastrointestinal tract in this role of digestion, breaking down big insoluble molecules into small soluble molecules ready for the process of absorption.